Go.
Okay. Welcome to Chemistry 499 Fall Semester 2013. The first seminar is by Jake DeRousse, and it is entitled, right there, you can read it.
Are inositol monophosphatase inhibitors a viable mimetic for lithium in the treatment of bipolar disorder? First, I'm going to start with an apology. As I was picking a seminar topic, I was debating between going into grad school for biostatistics or pharmaceutical science. So I'm like, I'm going to use a topic that, like medicinal chemistry-wise, to see if I'm actually interested in it or not. So I found an article about lithium, and I'm like, hey, just a straight-up element that's being used as a treatment to something, I'm going to do this. And then it turned out to be very little straight-up chemistry and a lot of biochemistry jazz that, yeah, so there. Obviously, most of us are just chemistry, have a chemistry background, so I'm going to go into the background of two important aspects of the seminar, which is cell signaling and enzyme inhibitors. To start off, cell signaling definition is binding of signaling molecules to receptors, which initiates a series of intercellular reactions that regulates virtually all aspects of cell behavior, such as metabolism, movement, or differentiation. This can happen from direct contact from cell to cell, or from like releasing a transmitter molecule type thing. There's three types of cell signals. Paracrine system, or signaling, which is a molecule released by one cell acts on another directly to a neighboring cell. And examples like scar tissue or blood clotting. The next one is an autocrine signaling, which is a molecule produces a signal and then it acts upon itself. My example is cell-mediated cytotoxicity, which I don't actually know what it is, but that was the example. And then the third type was an endocrine signaling pathway, which is the one that's actually going to be used here. That one, hormones are secreted from endocrine cells, which then circulate to act as on target cells at distinct sites. They're slower than the other two because the molecules actually have to travel before they act on their target cells. It's done by the intercellular transduction pathway, which is just a chain of reactions that transmit signals from the cell surface to a variety of intercellular targets. We're going to kind of glaze over what the IP3 pathway is, which is the one that is specifically used, or that they think is used in lithium. It's a second messenger, which just means it's inside the cell, as opposed to like hormones or things that act outside the cell. It's a product of phospholipid hydrolysis, which this is an example of pretty much just cleaving off an R group off of a phospholipid. And the end product of this pathway, specifically, is to release calcium from the endoplasmic reticulum at the end of it. It starts with G protein coupled receptors, which I had no idea what they were before starting the seminar. G proteins are guanine nucleotide binding proteins. They're heterotrymic G proteins. They turn on effector molecules, which the effector molecules essentially are the ones that make the second messenger. In this specific case, they turn on phospholipase C, lipase C, I'm assuming that's how it's pronounced, which then in turn cleaves PIP2, which makes phosphoinositol and PAG, which I have later on what that actually is, but we don't study that one very much. Here it's diacylglycerol. Then the inositol 1,4,5 trisphosphate, which is the ion base, I believe. It's a small polar molecule that is released into the cytosol, which is just the liquid found inside cells. After it's released, if its levels increase to 1 micromolar, it affects the activities of target proteins. Then once it increases the cytolic, that calcium, once that gets to about 0.5 micromolar of the concentration, the calmodulin is activated. It kind of acts to mediate the effects of the calcium inside the cell. There's what's happening, essentially. Stay on that. The next concept, biology concept that a lot of us probably don't know here is inhibitors. They're used to slow down enzymatic reactions, the rate of the reactions really. There's three main types of inhibition: competitive, non-competitive, and uncompetitive, which seems kind of confusing. Non-competitive, uncompetitive. This is when I'm drawing on my board. Competitive makes the most intuitive sense. It's when an inhibitor competes with a substrate for the active site of the enzyme where it's binding. So if this is an enzyme, and these are inhibitors, and these are the substrate, they're both trying to bind at the same place. So obviously if the inhibitor binds there, then the substrate can't because it's already taken up. So just by the inhibitor binding, say only half of the inhibitors bind, then there's only half of the usable enzymes left. So there's obviously less viable ones, and it's going to slow down the actual process. This one can be reduced, or the effects can be like reduced, and it can go back to normal if you add more substrate, which is unlike the other two. A non-competitive, it binds. Draw it again. Here's my enzyme. Inhibitor, substrate. Inhibitor binds at a site other than the action site, and it kind of changes the conformation of the enzyme. So then the substrate can still bind here, but it might shift it a little, it might do something else. It just, even though the substrate can then bind, it's not going to form the product. It can't release the product after that. Adding substrate to this one will not change the effects of it. And the last one is an uncompetitive, which is most interesting with me. In this one, we have our inhibitors might try and bind, but they can't yet. They can only bind once the substrate binds, then like another site might form, and then all of a sudden they can bind. So they can only bind after the enzyme substrate complex is created. Once again, adding the substrate does not affect the effects. Essentially, it can't go back to normal. In all cases, the reaction rates are basically reduced. Like I said, enzymes slow down the reactions. There's kind of just a graph of, I believe the blue is, it shows that 1 over S is the substrate concentration, 1 over V is the rate at which the substrate is like turned over. And as you can see, when we inhibit it, rate lowered case. And now on to actual kind of more chemistry, not just biology. General overview, lithium is the main treatment of bipolar disorder, but it has many adverse side effects, including like weight loss, and it's very toxic. The modulation of the PI or IP signal pathway is proposed as the treatment, what lithium actually does to create or to fix the bipolar disorder. We don't know for sure yet. This is done by inhibiting the enzyme IMPase. And as such, because of the adverse side effects, scientists have desired another inhibitor of IMPase and decided that could be a mimetic or an alternative. It mimics the effects of lithium. So as I was putting this together, it really has no benefit of the thing, but I was interested in how lithium was found. Jose Bonifacio de Andrade y Silva discovered pizzolate in the 18th century in Sweden, just a stone that had lithium, didn't know exactly what was in it yet. Then Johann August af Frentzen found lithium, like he specifically said, okay, lithium is in here. in 1817. And then later, finally, W.T. Brande and Sir Humphrey Davy isolated lithium in 1815 by electrolysis of lithium oxide. And lithium actually comes from the Greek word lithos, just meaning stone. So kind of a boring name. But. Lithium and bipolar disorder. John Cade he was an Australian psychiatrist, found the anti-manic properties of lithium. In 1949, it was actually completely 100% a mistake that he found this out. He thought urea could actually induce mania, so he injected lithium urate into guinea pigs to see what it did, and it actually made the guinea pigs sloth-like and lethargic, I believe was how they termed it. So then he's like, well, maybe the lithium reduces mania. So he treated manic patients with lithium carbonate and had very successful results with it. So it was proposed and has been accepted that lithium does treat bipolar disorder. Previous uses of lithium were just as a use to treat gout or as a salt alternative to people with heart problems. And still today, over 60 years later, lithium is still used as the primary treatment for bipolar disorder, even though it has all of its lovely side effects. Those side effects, besides the toxicity is more, it's prescribed at a 0.5 to 1.3 millimoles per liter of blood therapeutic window, and overdose can happen at just twice the therapeutic dosage, which is obviously really bad. So accidental overdose it happens a lot more than it really should. So obviously, we're in the 21st century, we shouldn't be having drugs that actually kill you. Kind of go in what lithium might do. It's not as important as finding the actual inhibitor, but it attenuates the IP signaling pathway, like I said, done by the inhibition of IMPAs. The mechanism is still unclear. It could be by the modulation of G proteins, or it could be by the regulation of second messengers, and that option is more attractive, so that's one scientist pursued. The effect of the inhibitor is more pressing than what lithium actually does. We know it works, and we want to mimic it, but don't know exactly why it works. Some indirect evidence links it. Two tests that were done specifically. Some peripheral cells of bipolar disorder patients were looked at, and they found out that they showed that the calcium levels, just the resting calcium levels, were a lot higher in bipolar patients, which kind of strengthens the theory that the reason that this happens is because the IP pathway is overactive and it's producing too much calcium. So then there's too much there. Another one is a 31P PChem crap that I'm not going into today at all. Magnetic resonance spectroscopy was looked at the brain levels of patients, and it also showed that the phosphomonoesters inside of the brain were increased, and also that the calcium levels were increased. So then there was a few tests done on non-humans that also strengthened. Allison, who was one of the authors in one of my two main lead articles, he observed the levels in rat brains and showed that levels decreased when you added levels of the inositol, we don't want decreased, and I1P, which is inositol 1-phosphate, another type of inositol, but it increased, which is what we want. Another test of cell cultures of rat brain sliced models showed that there was an accumulation of cytidine monophosphoryl phosphatidate, which here on out will be known just simply as CMPPA, that's a co-substrate along with inositol for the IP pathway, and also myo-inositol, which is just a natural forming version of inositol when it is added to a system, I guess, that's already been fixed by lithium. It shows that it counteracts the levels. There's no more accumulation of the CMPPA and the inositol levels are actually increased, the ones that we don't want. Lithium has a very wide range inside the body. It has a lot of uses. It also has other aspects in the signal transduction pathway. It inhibits cyclic AMP levels, which is just it alters G proteins that stimulate the catalytic conversion of ATP to cyclic AMP. It also decreases the increase in the binding of GTP to G proteins. So, okay. Now the ion pace inhibitors, they broke it down into three main types when they were trying to study to find if any of them were non-lithium cations, natural product inhibitors, and substrate-based inhibitors. The non-lithium cations, they know that ion pace requires magnesium to support activity. So, like that's substrate. Other cations that compete with magnesium for binding sites on the enzyme can also inhibit the ion pace. They use specifically calcium 2+, zinc 2+, manganese 2+, beryllium 2+, and gadolinium 3+. They are all competitive inhibitors of ion base, whereas lithium is a uncompetitive one. They prevent the magnesium-dependent hydrolysis of the substrate, whereas lithium inhibits it after the hydrolysis. This result mainly shows that the type of inhibition doesn't matter at all. It just, the rate needs to be decreased. And then natural product inhibitors. This one, I don't really know what made them think, let's choose this, but they isolated it from Memnomiella echidnata, which is that grape-looking thing there. It's a fungus found in cotton and wool. Specifically from that, they derived a compound also with a great name called L671-776. Great name. It's right there. It's a sesquiterpene with a triprenylphenyl. Anyway, these, they also found another one called K76COO-Na, but the Na was just, the Na is sodium, so there is actual still chemistry there. These inhibitors have a non-competitive inhibition, which shows that the mechanism is not dependent on the interaction of the substrate or like when it binds or anything like that. And then finally, they only had these two. They wanted another one, like a competitive. They wanted a competitive inhibitor to try and test, and so they did substrate-based inhibitors, which is what this. I decided to add the synthesis to it to try and add a little chemistry. I say synthesis because when I'm talking to Dr. Armstrong, we both decided that it's not like from here we add one thing and we get here. It's, well, we did this one, and then we wanted this, so we just use this one. So it's kind of a synthesis, but at the same time, it's not one-step synthesis. So we started with the inositol 1-phosphate, which is here. That was one of the substrates we were using. From there, I mean, it's an inhibitor. We already know that. So we just wanted to do some hydroxyl deletions. So they did a bunch of different hydroxyl deletion analogs, finding which ones were better than others. They found that the 2 and 6 spots were important for substrate binding and enzyme mechanisms, respectively. They found that this one, which is 2,4-dihydroxyl inositol 1-phosphate, was the best of all these analogs. So they used that. It has better affinity than the original one. So from here, there was 2 different paths that they could choose. The first path was just to add a bulky group on here. It was more potent than the original one also, but there was nowhere they could really go from this compound, so it kind of didn't pursue anymore. on that. So then the second pathway was just to replace the phosphate here with a bisphosphonate group. And that's, there's, like I said, all these names are really they were kind of annoying. I wanted cool, long names. But anyway, it gave this compound, which had better affinity than the others. Then we added a methyl onto bisphosphonate, the alpha position. Once again, all these, like as it's going over, we're getting more and more affinity, even if it's not very much more, but it is improving. From here, they just wanted to change the cyclohexanediol part to a phenyl part, which was kind of the step where we're like, okay, this wasn't just a one-step thing, because you can't really do that just one step very easily. It's impossible. But anyway, this got rid of stereospecificity, as you see. This hydroxyl is pointing out, that one's pointing in, and these ones are just flat, all on the same plane. So from here, we took off the
hydroxyl and got L690-330, which turned out to be the best, the optimized position. We actually deleted that to, we deleted it and it allowed it free rotation and the optimal binding, which is why it was the strongest one. But it turns out that this compound has poor cell penetrability. It can't really get in the cell. So they converted it to L690-488, which got rid of the charge group that's in this one. That one was actually able to get into the cell, and then once it's inside the cell, it's supposed to cleave off an ester that they added, and then converts back to this optimally. Like that's what they wanted to happen. Now all of these, they also had to look at the biochemical properties of the inhibitors. Like the point of this entire search for a new inhibitor was we want one that does what lithium does, but doesn't affect anyone else. So the non-lithium cations were found to be very effective. Most of them actually worked better than lithium at actually inhibiting ion pace. So this was good, but it produced many psychological side effects and also produced toxicity, which is exactly why we don't want lithium. Now they still are used to try and find the mechanism and structure of ion pace, like in tests, but they can't be used inside a person. Then the natural products also found to be effective. One of the tests that they did reduced pilocarpine-induced seizures. That's pilocarpine. It's a compound found from plants. Now this is another like effect that we know is modulated by the inhibition of ion pace. They use this because scientists can actually induce seizures, whereas you can't, or at least we shouldn't or don't know how to induce mania. So they use that, and it's found that the natural product inhibitor does inhibit the ion pace and reduce the seizures, but they have so many different uses. The natural products ones do, or like they affect so even more inside the cell than lithium does. So we can't really say, yes, it was done by inhibiting ion pace, so it didn't really help any. So then back to the substrate-based inhibitor, the L690-330 and the L690-488. So the L690-330 was found to be 500 to 1,000 times more potent outside the cell, than lithium was. But then when they tested it inside Chinese hamster ovary cells, it was found that, I don't know why that, but it was found that it actually was much, much less potent because very few of the compound actually got into the cell, as mentioned before, so there's less of the inhibitor to actually compete with the substrate. So then they used the L690-488, and it was more potent. It had higher of the CMP-PA, which is actually an indication, it's an index of the reduction of intercellular inositol levels. Since the results, like the L69488, it inhibited or it had similar levels of CMPPA to lithium. Said that it could work, but we don't know for sure. This is kind of a indirect proof of why lithium does what it does in bipolar disorder patients. There's a few problems still with this, with these theories. There's no conclusive evidence that the INPACE alone causes effects on the pathway, which I mentioned before. There's only indirect evidence. I mean, we think that's what happens, but we don't know for sure. And then there's a paradox that actually exists. Charged, I'm just going to read straight from what they had in the paper, but charged group analogs analogous to the phosphate group of the substrate are required for the compound to bind at the active site. This the polarity limits the bingeability. So, like I said, we wanted the L690-330 compound is what we wanted as the inhibitor, but it couldn't get in. And then the L690-488 can get in, but it's not going to inhibit what it does. It cleaves off an ester to get to L690-330. But we don't know for sure that that's what happens. That's what we wanted to happen. That's not what actually, per se, happened. So what now? Say when I initially decided to do this topic, the goal, or what I thought was going to happen, because it was in a very new journal, I thought it was going to say, okay, we have this new drug, and it's now being tested. It's on clinical trials, and here to learn about it. And instead, it was, big long art sent me to a bunch of different articles about these inhibitors, and then says, well, we still don't really know why. We still don't have an answer. So they said that the nonpolar, what they think could work is if you got a nonpolar inhibitor, so no charges, it can get in, that can interact with hydrophobic parts of the enzyme, but first you have to figure out what those are. So really the next step is to find out the hydrophobic parts of the enzyme and then develop a inhibitor to interact with those parts. So they don't know yet. They're hoping that they soon will. And now I'm ready to get ripped apart with questions. So the floor is open. Yes.
Excuse me. You may have mentioned this and I might have missed it. Lithium toxicity.
Yes.
What makes lithium toxic? I know why a lot of other things are toxic, but what is lithium toxicity attributed to? The reason while you're thinking about it, the reason I ask that is there are things that might be toxic. There are a lot of drugs that are toxic. Strychnine, digitoxin, the dose makes the poison. I'm just curious as to what lithium does. Potassium, for example, is part of the Kevorkian cocktail, overdoses, and just wondered if you ran across that. And the reason I'm asking is some way to mitigate that, maybe.
Yeah. Yeah, I did remember reading that they tried to, actually, after having an overdose of lithium, try to reverse the effect if they can. But I don't remember specifically reading it. I would hypothesize that since in this pathway that I was talking about, we act like it serves a purpose, so releasing the calcium, we want to do that, and just the bipolar disorder is caused by releasing too much of it, that if we have too much lithium in there, then it will then inhibit too much, it will inhibit too much of what we actually want, and then maybe that could, I don't know exactly what function it causes, but maybe it just by having too much in there, it will inhibit it too much.
Lithium affects the release of calcium?
That pathway releases calcium, and lithium inhibits the pathway.
Okay, so then that would mean less calcium?
Yes.
Okay.
Less calcium released from the endoplasmic reticulum.
Okay.
Or the ER, which made it sound funny to me.
Calcium necessary for smooth muscle?
I remember reading smooth muscle traction, I
Yeah,
think.
right.
So, yes,
Okay.
but I don't know exactly
why, I just know that they want that pathway in normal people. I mean, it makes it up to 1 micromolar concentration.
Sort of related question about lithium positive ions. Looking at the periodic table, of course, they're going to be tiny because lithium has 3 electrons. If you take away 1, you're down to 2 electrons, so you just have a filled 1s orbital. So I would think that lithium plus ions are way smaller than any other ions on the periodic table, except possibly beryllium plus 2, which would also have a 1s2 configuration. So I'm curious as to why they thought that big ions, I think what you call it, non-lithium ion inhibition, yeah, like a gadolinium plus 3 that you mentioned is going to be huge, having what, like 61 electrons in it. So I'm guessing that the bigger ones, they're not assuming that the bigger ones are replacing the lithium in the active site. It must be a non-competitive
Yeah, those
inhibition.
ones I believe I said were non-competitive, so it's not.
Because I would think that if there's a binding site for lithium, it's going to be just tiny so that the lithium just fits into it. But bigger ones wouldn't fit into it, so it must be non-competitive.
Was the lithium competitive?
The lithium was not competitive. It was these ones, the non-lithium ones were competitive, and then the lithium one was, I think, uncompetitive is what it said. I read it in there sometime. So it was one of these.
Space is big enough for the other ions, and lithium does it a different way.
Right.
The lithium doesn't. The lithium isn't specifically competing with the substrate because, like you said, I mean, I don't know if they themselves really care how big it is, but it's so small that maybe it's not going to affect it as much if they're competing. But it's one of the. I think it was not competitive, which was the second one, which means that it's binding to a different place, essentially.
And then I guess the same thing would be true for these various organic ones because they're nothing like lithium.
Yeah.
They must be binding to a different site, maybe. Or
One of the kind of attributes of a competitive inhibitor is that it looks like the substrate,
right.
So it can get into the same places that the substrate would be. So the substrate-based ones obviously are also competitive.
Right.
So also the same as lithium. Actually, none of the inhibitors that they tried were the same type of inhibitors as lithium. We don't exactly know why they didn't try and find one that had the same type of inhibition, but they didn't talk about one.
Well, I think the problem is partly because lithium is so different from anything else. It's going to be hard to find analogs for it.
And like I said, even back when it was initially found that it worked, they didn't really. It's not like they thought, hey, we should try lithium to treat this disease. It was just a happy accident.
How big is what they're inhibiting? Is that the enzyme?
The iron base, entire section on the structure of it that I did not go over for this presentation at all.
Is it macromolecule size? Is it polymer? I mean, how?
It's an enzyme, right?
Yeah.
So it's huge.
I mean, I assumed it's big, but I don't actually know how big at all.
One of the, along the same lines, you have lithium can increase the levels of dopamine. Of course, that's one we're all familiar with. But in the range of 32 kVA, is that kilodaltons?
I think it was, but when I was talking to Dr. Reed yesterday, he had it circled. So, and then I went back and looked at it, and I still think it's kilodaltons, but I'm not.
I thought Dalton meant related to molecular size as opposed to concentration.
I mean, I might have miswrote it.
Kilodaltons, aren't they more size, right, as opposed to concentration? Okay. Well, because he used it to increase the levels of dopamine to 32 kilodaltons.
There was many a typing or logic error in my
I'm not
rough
criticizing that, Jake. I was just interested.
Yeah.
I thought maybe you were talking about when you said the levels, that there's some kind of change in molecular size or something. Okay.
I think they're related with slightly different question. How do they tell how good they are at inhibiting? What do they do?
Well, One, I mean, like I said, the main way that they test how good it is just to see how much the resting levels of calcium in the cytoplasm within the cell, I think is the
So
word.
it's a result-oriented, not a process-oriented
Yes.
test.
Yeah, so they pretty much just, they want us, there's a way that they can see how much calcium is not binded to something else, I guess, not binded besides its counteract, because it's
Free calcium.
So they take it, they apply whatever chemical they're testing to it, and then they just test the free calcium at the end.
That's what the main way was. I mean, I think there was other ways to test it, but they didn't go into that one as much.
Do you know offhand what kind of tests they did to test the calcium? Was it some kind of a,
Well, that
Spectra.
magnetic spectroscopy thing,
oh, okay.
1P orbital splitting stuff. Travis will talk about next week.
So you're up,
Maybe.
so you're actually measuring the bound calcium by measuring the phosphate levels that are bound versus unbound?
Sure.
Okay.
Well,
If you're using phosphorus NMR, you're actually not looking at calcium, you're looking at phosphorus levels of different types.
there should know what the significance of the 31 is, Jake. There's probably, I wonder if are they using spectroscopy, you know, like atomic absorption or flame emission or something like that?
Didn't talk about that at all. The one thing that I don't remember if it was for this when they were looking for the structure of the ion pacer when they were seeing if it worked. They talked about using mutagenesis, which I'm not at all an expert as to what it is, but I remember that as a buzzword as to something they did afterwards. So like I said, I can't remember if that's when they were trying to figure out, because they have tried to figure out the structure of IMPAs just because knowing the structure of it will help them find an inhibitor that will actually work for it.
When they were going through all that, I'm assuming by how you talked about it that it was simply steric binding effects. They didn't look into electronic effects at all.
If they did, they didn't talk about it at all. They simply said, they didn't talk about the synthesis very much at all. And I was a little disappointed in it, but it was pretty much. I think they just tested a bunch of different things, and then the ones that worked, they put in and said, "Yep, we went from here to here." I don't know if they really cared as much what they were doing. They might have tested all different types of changes, and then others didn't affect the affinity as much, I guess.
One thing I think you should be aware of, everybody, is that the synthesis that's on the board, is, as Jake correctly said, is not a synthesis at all. It's basically a thought process. If you know that first molecule is an inhibitor, then they thought, well, maybe what if we come up with a molecule that is missing 3 of the OH groups? It doesn't mean that they made it from that original compound. It means they're just basically showing what is the thought process and how do you get more and more active inhibitors by changing the molecule somehow. So they probably actually synthesized the second molecule totally independently of the first one. It's just that's the thought process for why are we going to try that second molecule. Okay. But I do have a question about the scheme that's on the board, and that is, you said that L69488, which is, I guess, the really super good one, has some kind of group on the OH. Do you know what the group was? Did you mention that, or?
I didn't mention that. I don't think it was.
You said it's an ester of some sort.
Yeah, it was a really long name. I'll find it for you somewhere. And now I wish that I would have actually kept this in order. But
And the idea, I guess, is that the free OH group on that para position somehow blocks crossing the cell membrane. But if you put another group on there,
Well,
it makes it.
I was gonna ask that too, because he said it was something about a negative charge, but I don't see a negative charge on the 69330. And he said it was the negative charge that kept it from getting into the cell.
Right. That's partly the thrust of my question.
I assume negative charge. Well, these are just, you said bar and the thing has some negative charge there. You said it in the this phosphate group is where the negative charge was. The thing that they did,
So
they
it's
added.
a local charge, not the entire molecule charge.
Yeah, but at the same time, it's still any charge whatsoever makes it not be able to sell
We
it.
had the sodium salt,
Right.
phenoxide, if you want to call it that.
Right, that's what I thought you meant, was that the OH group at the bottom, because it's phenol, I guess at physiological pHs would have something of would actually be its conjugate base, and therefore have a negative charge on it. But if you esterified that, then you wouldn't have a negative
No.
charge. So, again, I'm asking if you know what the structure of 690, 488 is.
It said that they added a tetra p-val. I'm gonna write it down because I can't pronounce that one at all. And they wrote it. They never showed what this one was, but. Tetra.
Oh,
Ester. They added that, or made it into a that. One of those.
okay, so, yeah, that's a big organic group, I could probably figure it out, but pivalonic acid is something, so that's based on 4 of those apparently. But is that attached to the OH group on the benzene ring? Or is it attached somehow to the phosphates? Attached to the
One of the
phosphates?
oxygens.
Oh, okay. So the negative charges are on the oxygens of the phosphates, and you're essentially deleting those negative charges to make it able to cross
Yeah.
the membrane. Okay, that makes sense.
That's why once it gets in, because we know that this is the best, whatever, so once it gets in, then we can complete that one back on.
Okay, that makes sense. Students, do you guys have any questions?
There's one part you said, I wasn't quite sure what you said. You said when they went from at the very end, you were talking about the different abilities of the 330 there and the 488. So the 330 can't get in as well as the 488. Plus, then did you say something about the 488, once it gets in?
488 isn't as
breaks
good.
down to the other one?
The 488 doesn't inhibit it as well. So they more optimally, like in an optimal world, ideal world, it would then cleave off the ester. We don't know exactly that's what happens. That's what they want.
Cleave off the ester, and which
The
would
one
make
the
it go
thing
back to
that-
the 330.
Yeah, which puts it back to this. They want it to get in and then go back to the one that we need. They're just trying to get around the fact that this one that we know works really well can't get in.
So they want to get it in and then find a way to convert it back to what it was. Is that common doing reactions inside the cells? Is that was important to me.
Yes, it's very common, I think.
I thought that's what you said, it just, I wasn't sure.
So they want it to happen, they don't know what happened. That's why we don't have this as being the new lithium type drug, bipolar disorder drug, because we don't know it doesn't happen.
If, you said they tried a lot of different metal ions as inhibitors, and you said that a lot of them worked, but they had other kind of side effects and things. What I was wondering is, many drugs use various metals as cations, I assume. How come they all don't inhibit this then, if they found so many different metal ions that will inhibit it?
I mean, they just talked about the ions, they didn't talk about necessarily what went in with it. That might have something to do with it or how you inject it. I just like, you said, yeah, I'm assuming a lot of medicine does drugs use those same ones, but I assume it's all has to do with how they actually treat it.
Lithium, okay, besides the brain activity, is it important for any other cellular processes elsewhere in the body? Because magnesium and calcium are, and maybe that is the issue that
The main ones that they ran into were those two ones also in cell signal, the conversion of GTP binding of G proteins to GTP, which is just based in energy, and something cyclic energy. So other pathways, other parts of the pathway, but those weren't as important for the basis of this study.
From a physiological point of view, lithium's not used hardly at all, I don't think. I can't think of one spot in the body where lithium is used as a cofactor or a coenzyme or anything. Maybe it is, I don't know. It doesn't mean it isn't.
And none of the articles I read mention anything about, and it also does this.
Right. I just had one little teeny tiny question. At the very beginning of your presentation, you talked about different kinds of crins, autocrine, paracrine, endocrine. And you talked about the last one, the endocrine system. And those are hormones that cause the signal transduction pathway. And then you said something about in relationship to this, going back to your 330 and 488, the issue of getting across the cell membrane. That's an issue, right? The failure of 330 to get across. You changed it to 488. Are there other hormones that can get across the plasma membrane that you can think of? Or do hormones ever cross the plasma membrane? Or do they only do signal transduction with the second messenger? Good question.
I remember you said that testosterone was counts as a hormone, but I know that was one of them that is used in the pathways, and I'm pretty sure it's in.
So it can cross the plasma membrane?
But
Testosterone can cross?
I believe so. Yes. Okay.
Yeah, absolutely.
I'm pretty sure.
Even though it's really big.
That was the example. We
It's
used
like
to-
cholesterol.
That example.
Yeah, it's very hydrophobic and it can cross the plasma membrane.
What about insulin?
Can insulin cross the plasma membrane?
Insulin was actually the example that they showed before them. I'm going to say no.
Correct again. I'm done. 2 in a row. Okay.
Well, it makes sense chemically if you think about it, because apparently nonpolar things are crossing the membrane fairly easily, and testosterone being a steroid would be fairly nonpolar. But insulin is a
Amino acids.
peptide,
Right.
yeah, which means it's bipolar, so it's going to have trouble crossing. That compound, 330, is quite polar because of the charges on the oxygens of the phosphate groups.
How does
But if you
regulate sugar?
neutralize the negatives, then it becomes nonpolar, and so it crosses the membrane. So it makes sense.
Yes, Paul.
Why does lithium taste like salt?
Does lithium taste like salt? It is a salt.
Well, yeah. Okay.
Show me your non-chemistry answer the question. Or don't.
Pass? Okay.
That's not right. I guess it's tricky.
The salt taste comes from the cation. The chloride just kind of goes along for the ride. I'm sorry, the chloride gives the taste. And the cation just goes along for the ride. They taste different, but they have the effect of making things feel salty, taste salty.
I've never had lithium salt, so.
Have you eaten lithium, Paul?
Not.
How do you know it tastes like salt?
Fully commit to this.
Because it was used as a salt replacement for people with.
Yeah,
Gotcha.
that would have been an interesting
Good question.
Paul
research.
thought bipolar disorder was missing a chance to go to both poles of the.
Well, we're out of time, so let's thank Jake.